Aim. To understand the structural components and their functions of a thermocycler, PCR machine. Apparatus/materials required. Thermocycler, PCR machine, PCR tubes, sample DNA, primers, nucleotides, and DNA polymerase, power supply. Theory. A thermocycler, also known as a PCR, polymerase chain reaction, machine, is an instrument used to amplify DNA through repeated cycles of heating and cooling. It was invented by Kary Mullis in 1983 and is one of the most important tools in molecular biology and biotechnology. The thermocycler automates the process of denaturation, annealing, and extension, enabling exponential amplification of a specific DNA segment. Precise temperature control and rapid thermal changes are essential for accurate DNA replication. Structural components and their functions. Part. Function. Thermal block/sample block. Holds the PCR tubes, heats and cools rapidly to achieve temperature changes for DNA amplification. Usually made of aluminum for efficient heat transfer. Lid/heated cover. Presses tightly on PCR tubes to prevent evaporation and condensation during cycling. Temperature sensors/thermoelectric modules. Maintain precise temperature control and uniform distribution across the block. Control panel/keypad/touch screen. Used to set parameters like number of cycles, temperatures, and durations for each step. Display screen. Shows the temperature profile, running time, and cycle progress. Microprocessor/controller unit. Regulates heating, cooling, and timing based on programmed protocols. Cooling fan/ventilation system. Prevents overheating and maintains stable internal temperature. Power supply unit. Provides required electrical energy for heating and cooling operations. USB/data port, in modern models. Allows saving and transferring of PCR programs or data. Housing/outer casing. Protects internal components and provides insulation. Working principle. The thermocycler works based on the principle of temperature cycling, which allows DNA amplification through PCR. Each PCR cycle includes denaturation, 94 to 95 degrees Celsius double-stranded DNA separates into single strands. Annealing, 50 to 65 degrees Celsius primers bind, anneal, to complementary sequences on the single-stranded DNA. Extension, 72 degrees Celsius to DNA polymerase synthesizes new DNA strands. These steps are repeated for 25 to 40 cycles, leading to exponential DNA amplification. Procedure. Prepare the PCR reaction mixture, DNA template, primers, nucleotides, to polymerase, buffer. Dispense the mixture into PCR tubes and place them in the thermal block. Close the heated lid to prevent evaporation. Program the thermocycler with desired temperature cycles. Start the run and allow it to complete all programmed cycles. After completion, remove the amplified DNA samples for analysis, example gel electrophoresis. Observation. The thermocycler runs through programmed temperature cycles automatically. After completion, a large amount of DNA is amplified from a small initial quantity. Conclusion. The thermocycler, PCR machine, is a crucial instrument in molecular biology that enables DNA amplification by automated temperature cycling. Its components ensure precise heating, cooling, and timing, making PCR accurate, efficient, and reproducible.